Hi all, and welcome to this lecture on antigen presentation. As you probably remember from prior lectures in the seminar series, all the figures that are referenced in the Janeway textbook will also have their numbers listed in the upper or lower corners as a way for you to follow along using your textbook. And this can be helpful as some of the figures might have writing that's kind of small when viewing it on these particular PowerPoint slides. So that's just a way for you to follow along. All right. So I would like to start out this lecture with some primary objectives. These are the things that we hope to learn over the course of the lecture. And so for our first primary objective, we want to understand how a T cell can recognize its cognate antigen and how this will differ from B cells. I know we haven't learned about T cell receptors or B cell receptors yet, and so we're getting a little bit ahead of ourselves in trying to understand this mechanism of antigen presentation, but in the next lecture we will be learning about lymphocyte antigen receptors, so you'll have a better handle on why this is important once we get there. Additionally, we want to understand these molecules that are referred to as major histocompatibility complexes, or MHC, and there's an Mhc Class 1 and 2, and we want to understand how they work and why they're different and what their different functions are. We also want to know the cell types that can present antigen to T cells on Mhc Class 1 or Mhc Class 2, essentially forming the link between the adaptive immune system and the innate immune system, which you've talked about in prior lectures. We also want to understand this process referred to as cross-presentation, and we will talk about that process over the course of the lecture. All right, so starting out, I want to introduce this concept that's called Mhc Restriction. So as mentioned previously, antigen presentation is going to rely heavily on these molecules referred to as major histocompatibility complexes, and this is often shortened to MHC. And so for the rest of the lecture, I'm pretty much going to refer to them as MHC. And there's a Class 1 and Class 2. And the reason that these are important is we've learned a lot about the adaptive immune system. So here we see a dendritic cell, which is we've learned a lot about the innate immune system. We have yet to learn about the adaptive immune system. So right here, we have a dendritic cell, which is part of the innate immune system, shown here. And as we know from prior lectures, when a dendritic cell or a macrophage interacts with, for example, a bacterium, it will take up that bacterium as part of its response. But what we have yet to cover is that dendritic cells in particular have a way of communicating with T cells, which are part of the adaptive immune system, thereby providing the link between the innate immune system, which is essentially the first responder in terms of infection, and communicating the information to the T cell about the type of pathogen and about the type of responses that should be had. And the way that this is accomplished is once the bacterium is taken up, then the antigen presenting cell, in this case, which is a dendritic cell, will use the MHC molecules to interact with T cell receptors. And so this will communicate to the T cell about what kind of pathogen there is and about what kind of response should be generated in order to effectively have an adaptive immune response. And so this brings into question the difference between B and T cell antigen recognition. So if we look at our B cell receptors, they actually will interact with native antigens, so they do not interact with MHC at all, and they will just see their antigen floating around and interact with it through their B cell receptor. By contrast, T cells, which come in two different flavors, which we'll talk about in more depth, there are Cd4 T cells and Cd8 T cells, and for them to interact with their antigen, they need to see it presented on these major histocompatibility complexes, or Mhc Class 1 and 2, which are presenting the antigen as part of the innate immune system. So, as mentioned before, we can see a B cell receptor over here interacting with a native antigen. And when I say native antigen, that just means that it's taking on its three-dimensional conformation. And so it's not stretched out in any way, it's folded, and that is the way that the antibody is interacting with that particular antigen. Conversely, this process that we'll go through over the course of this lecture in understanding antigen presentation on MHC shows that T cell receptors actually are only interacting with antigen that is in this peptide form that is presented on the MHC molecule, which is present in antigen presenting cells such as dendritic cells or macrophages, but also can be present on all cells of the body as a way to alert T cells of the initial stages of an infection. So this is critical communication, but this is a major difference in the way that B cells will interact and recognize their antigen, and T cells will interact and recognize their antigen. So as mentioned previously, and we'll get into the functional differences of these in more depth in subsequent lectures, but I just want to help to contextualize the different types of T cells before we get into what is happening during antigen presentation. We have two different types of T cells that are differentiated based on the presence of specific co-receptors. And so in one T cell subtype, there are these CD8 co-receptors, and this will facilitate interaction with Mhc Class 1, as we'll come to see. And then there's this other kind of T cell which has the co-receptor for Cd4, and this, as we will come to see, will facilitate interactions with Mhc Class 2. So for our CD8 positive T cells, it's important to remember that these T cells are going to respond to intracellular pathogens, such as viruses, and this will help to explain why antigen presentation happens the way it does for Mhc Class 1. And so, as mentioned previously, these CD8 T cells are going to see their antigen when it is presented on Mhc Class 1. By contrast, Cd4 T cells are going to respond to extracellular pathogens, and these extracellular pathogen antigens are going to be presented on Mhc Class 2. And so it will make more sense as to why antigen presentation happens a certain way for Mhc Class 1 and Mhc Class 2 when we understand the types of T cells that they are interacting with and what the purpose of those T cells are in an immune response. So just summarizing this a little bit more, we have our CD8 T cells, which during an immune response, when they're stimulated, will produce these cytotoxic T lymphocytes, or CTLs, and this will be discussed in much more depth in subsequent lectures about immune responses, but this is just another way to contextualize. So we also have the CD4 T cells, and these will become, during an immune response, helper T cells. So the primary job of these CD8 T cells is going to be killing infected cells. So as mentioned in the prior slide, the CD8 positive T cells are interacting with virally infected cells. So in the face of a viral infection, the immune system responds by eliminating the infected cells as a way to try and contain that overall infection. And so CD8 T cells will mediate this response. So the information that is needed here that is presented by Mhc Class 1 to the T cell is a sign that the cell is infected. And so this will activate the CD8 T cell to kill virally infected cells if viral peptides are presented on Mhc Class 1. Then when we look at our helper T cells, ultimately, their job is to assist in an immune response. And so here, they're generally shaping the immune response to fit the particular pathogen that the organism is infected with. And so here, information that needs to be conveyed by Mhc Class 2 to the CD8 T cell is the type of infection and how best to respond. So the different types of pathogens that an organism, a multicellular organism, can interact with are these cytosolic pathogens or the intracellular pathogens. There are also intravesicular pathogens. These will be more common for our antigen-presenting cells, which can uptake, or the phagocytic cells, which can uptake particular pathogens, and sometimes pathogens have ways of propagating within these vesicles when they are phagocytosed, but oftentimes they'll leak into the cytosol. So we'll come to see that the cytosolic pathogens and the intravesicular pathogens will be responded to in a very similar way. And then we also have these extracellular pathogens, which will primarily be detected by dendritic cells or other APCs. Right here we see a B cell, and B cells will also respond and communicate with T cells, but this is all dependent on Mhc Class 2, as we'll get into. So first, starting out with Mhc Class 1, this is our mechanism of alerting T cells to an intracellular or cytosolic pathogen. So this is just shown right here. Before we get into how antigen presentation on Mhc Class 1 actually occurs, let's look at the structure of the molecule. So here we see Mhc Class 1, and it consists of this alpha chain, which is tethered to the membrane, and it also associates with this other protein called beta-2 microglobulin. So right here we see our alpha chain, which consists of an alpha-1, alpha-2, and alpha-3 domain. It also has this transmembrane domain, which tethers it to the membrane. And when expressed, it will interact with beta-2 microglobulin to form the stable Mhc Class 1. And we notice here that beta-2 microglobulin does not have a transmembrane domain, so it won't be tethered to the membrane like the alpha chain is. And when we look at the structure of how this actually interacts with peptide, here we have our Mhc Class 1, and this box is meant to show the peptide binding cleft. So this is where the antigenic peptides will actually interact with Mhc Class 1. And so if we take the Mhc Class 1 molecule and we flip it up and look at it top down, we can see this peptide binding groove, and right here is a peptide shown in yellow. And then the gray amino acids are those that are interacting with the peptide. Something that you can appreciate from this photo is that the ends of the peptide binding groove are relatively closed when it comes to Mhc Class 1, and Mhc Class 1 actually does have a preference for binding peptides that are 8 to 10 amino acids long. And so you can see how this is a little bit restricted, and this will differ from the interactions with Mhc Class 2, which we will come to see. So when we look at the genomic organization of Mhc Class 1, and this is for humans, we also can see that there is a different nomenclature for MHC when in the context of humans. So when it was discovered, people had actually named it HLA, which stands for Human Leukocyte Antigen. I'm going to continue to use the MHC terminology, but just know that if you encounter that type of terminology elsewhere, that HLA is the same thing as MHC. And so here we can see in the MHC locus that there are 3 different Mhc Class 1 loci. So there are going to be different HLA, A, B, and C. So there are 3 different types of MHC1, and they will actually have different specificities for different types of peptides, broadening the amount of peptides that will be able to communicate information to the T cells. So when we look at this process of antigen presentation on Mhc Class 1, I think that it's best to characterize this in 4 different steps. which we will go into in more detail. So the first step is antigen processing. This is the way of actually forming the peptides, and this will happen in the context of the cytosol. Then, because Mhc Class 1 molecules are synthesized into the endoplasmic reticulum, then the antigen will need to be imported into the ER in order to interact with Mhc Class 1, because it started out in the cytosol. For step 3, the antigen peptide will bind to Mhc Class 1, and this will happen within the context of the ER where this is all assembled. And then step 4, the antigen presentation pathway will commence via secretion during the normal secretory pathway that we're all familiar with. So going into these steps in a little bit more detail, we have step 1 here. So as we know, proteins can be synthesized on ribosomes that are either present in the cytosol or present on the ER, but this is just showing one that is cytosolic. And just keep in mind that this process is happening all the time. So when looking at this process right here, we're looking at the presentation of self-peptides on Mhc Class 1, but during an infection, you can also have presentation of viral peptides on Mhc Class 1. So we have synthesis of our peptide, and peptides, as we know, can be degraded through ubiquitination. So there are ubiquitination ligase proteins that will add these UBs to the protein, and this tags it for degradation. So once it has been ubiquitinated, it will move to the proteasome, in which the proteasome, which normally will recycle proteins and peptides within the context of the host cell so that these peptides can be used as amino acids to synthesize more proteins and etc., then they can be cleaved up. And so this is a normal process that happens in cells all the time, but it actually ends up being a crucial part of antigen presentation on Mhc Class 1 as part of the immune response. And so when we look at the proteasome, there are 2 different types of subunits, the alpha subunits and beta subunits. And something that I wanted to point out is that the proteasome is acting all the time within cells. So during autophagy, in which cells will recycle their proteins to make new proteins when they no longer need the old ones, this is a way for the cell to repurpose those amino acids. But in the case of antigen presentation on Mhc Class 1, this process of using the proteasome has actually been repurposed so that we can have the production of these peptides, which will eventually be presented on Mhc Class 1. And something else I wanted to point out, which we will get into when we talk about immune responses, is that there's actually a specific type of proteasome referred to as the Immunoproteasome, which is stimulated during infections. So it can be communicated to cells through the presence of different cytokines, for example, interferon gamma, during an infection to produce these other subunits that can be present in this proteasome. And the difference mainly here is that it will cleave peptides that are the appropriate length to bind to Mhc Class 1. So this provides peptides that are the preferred length, as we remember, which is 8 to 10 amino acids. So when moving into step 2, at this point, the peptide can be imported from the cytosol into the endoplasmic reticulum. So we have peptides that were generated by the proteasome, and then they make their way to the endoplasmic reticulum, but they need to have a way to actually enter. This is where this particular protein, referred to as TAP, comes into play. So TAP is this protein that interacts with peptides in the cytosol and allows for their transport into the ER where Mhc Class 1 is being assembled. So when we move to step 3, where we have peptide loading on Mhc Class 1, here we're in the context of the ER and we can see our Mhc Class 1 which the alpha chain is shown separately from beta-2 microglobulin, and at this state, before beta-2 microglobulin interacts with MHC class 1, we have this chaperone protein referred to as calnexin that is stabilizing the protein because it's not fully assembled. And then when we move over to this next panel, we can recognize this TAP protein, which we just talked about, which is necessary to move peptides from the cytosol into the endoplasmic reticulum, alongside of a whole bunch of other proteins, such as taposin, which is another chaperone protein that is facilitating interactions with MHC class 1 and keeping it stable. We also have calreticulin, which is doing the same thing. It's keeping the MHC class 1 stable. And something that you'll come to realize is that the stability is necessary because MHC class 1 is actually only stable when it is interacting with peptide. And so there are a lot of different chaperones that are keeping it together until it actually will bind peptide within the ER, allowing it to be fully assembled. And ERP 57 is also doing the same thing. So at this point, we can have peptide binding, and you can see that some of these chaperones are moving out of the way because now we have increased stability of the MHC class 1 once it's interacting with its peptide. It no longer needs to be interacting so closely with these chaperones. You'll also notice this other protein here called ERAP. It's an aminopeptidase, and the role of ERAP is to cleave down these peptides to make them the proper length of 8 to 10 amino acids so that it can interact with MHC class 1, because if they're too long, then they're not able to sit in that binding groove. Something else that's important at this point to understand about molecules such as taposin and ERP57 is that they will go back and forth interacting with MHC class 1 and interacting with MHC class 1 and constantly ejecting this peptide until we have a high affinity interaction of the peptide with MHC class 1. And so this is referred to as peptide editing, and it's a way to ensure we have a high affinity interaction of MHC class 1 with its peptide before it's presented on the cell surface. I also wanted to mention at this point that these 4 proteins right here, calreticulin, ERP57, taposin, and TAP, are referred to as the peptide loading complex. And so that's just some important terminology, and these are the proteins that are very important to remember that facilitate this peptide loading process. So at this point, we move into step 4, in which the MHC class 1 has been loaded. So we've had our peptides degraded by we've had our proteins degraded by the proteasome to form peptides, which can enter the endoplasmic reticulum through the TAP transporter. At this point, then they start interacting with MHC class 1, and once they have this interaction of MHC class 1, which is the alpha chain and beta-2 microglobulin bound to the peptide, then it will go through the normal secretory pathway through the Golgi network until it reaches the cell surface. So now that we have gone through how this actually happens, let's review the 4 major steps of antigen presentation on MHC class 1. So our first step was antigen processing. This is the process by which proteins are cleaved up into peptides by the proteasome in the context of the cytosol. After this, these peptides are transported into the ER by the TAP transporter proteins, and this is necessary because at first they were in the cytosol, but they need to be able to interact with MHC class 1 before MHC class 1 export, which all happens in the context of the endoplasmic reticulum. Then we have step 3, which is the antigen will be loaded onto Mhc Class 1 before its export to the cell surface. And then we have the last and final step in which Mhc Class 1 that's interacting with its peptide can be secreted to the cell surface and then interact with these CD4 T cells, which, as mentioned previously, will be important for responses to intracellular pathogens such as viruses. Now that we have gone through the process of antigen presentation on Mhc Class 1, I want to talk about how this happens for Mhc Class 2, and then we'll come back and make comparisons between these two processes. So something that's different with Mhc Class 2 is that this will particularly be relevant for the extracellular pathogens and toxins, which is mainly going to be for phagocytes and macrophages, as well as B cells, which are all antigen-presenting cells. When we think about Mhc Class 1, this can actually happen on all nucleated cells as a way to communicate to T cells whether or not they're infected. However, phagocytes in particular are those that rely on Mhc Class 2. So, as mentioned previously, Mhc Class 2 is responsible for the immune response against exogenous pathogens and exogenous antigens. So this is particularly relevant for phagocytes, which can take up exogenous antigens into their vesicles. And this will actually be the site of pairing the peptide with the antigen, as we will come to see. In later lectures, you'll come to appreciate that this interaction of Mhc Class 2 is particularly important for B cells as well as a way to communicate with T cells during an immune response, because T cells will help to inform and activate B cells. And so in addition to this being a way for T cells to interact with antigen-presenting cells such as dendritic cells, B cells rely on this as a way to communicate with T cells during an immune response. And so this the way that this will be facilitated is through recognition of extracellular or exogenous antigens through the B cell receptor, which will be uptaken and then presented on Mhc Class 2 to communicate with T cells. Before we get into the process of antigen presentation on Mhc Class 2, like we did for Mhc Class 1, let's go through some of the structural features of Mhc Class 2. So here we can see that Mhc Class 2 consists of an alpha chain, which has an alpha 1 and an alpha 2 domain, as well as a transmembrane domain. So you see this in blue in the picture, and this is what keeps it tethered to the membrane. Here it's being shown in the endosome, which is actually a very important site for Mhc Class 2 antigen presentation and processing, and so that's why it's shown here as opposed to tethered to other membranes. Additionally, there is this beta chain shown in yellow, which consists of a beta 1 and beta 2 domain, as well as this transmembrane domain. So one of the immediate differences that you can see between Mhc Class 1 is that there was only a single chain that was mediating the peptide binding groove in Mhc Class 1, whereas you have to have the combination of the alpha chain and the beta chain in order to form this peptide binding groove in Mhc Class 2. So when we look a little bit closer at this peptide binding groove, which is highlighted by this box right here, once again, this is a top-down view, we can see the interaction of this yellow peptide with the peptide binding groove, which is shown in gray. And another thing that should stand out to you as an immediate difference between Mhc Class 1 and Class 2 is that the ends of this peptide binding groove are not as restrictive in MHC class 2. So this can facilitate peptides that are a little bit longer, such as 10 to 13 amino acids, as compared to the preferred 8 to 10 amino acids for MHC class 1. When we look at the genomic organization, once again, so we saw our MHC class 1 molecules here. Here are MHC class 2 genes, and there are a lot of other genes associated with antigen presentation that we're not really going to get into today because they're extremely specific details. But what I wanted to point out is, once again, we have 3 different loci, DP, DQ, and DR. And so these are 3 different places where we can form 3 different types of MHC class 2. Once again, each of these will have slightly different specificities, and this will allow for maximum ability to present different kinds of peptides based on biochemical differences of those peptides. And something else you can notice that's different in the gene structure of MHC class 2 as compared to MHC class 1 is that there's a beta and alpha chain for each of these alleles, whereas for class 1 we just have that alpha chain because that's the one that's forming the peptide binding groove in the case of MHC class 1.